wing-shaped process called the pterygoid process. On a bone that has this wing to it and a second wing-like structure going up into the orbit like so, all of this is allospheноid. Allosphenoid. In front of it is another bone in the orbit called the orbitosphenoid. And the two bones, orbido and allosphenoid, encompass these four foramen, or holes. We have the optic canal, or optic foramen for the optic nerve. Slightly larger orbital fissure. Why don't we call it a foramen? Because in humans, where everything was named first, it looks like a fissure on a mountainside. This allows blood vessels to come into the orbit and service the retina, which is an outgrowth of the brain and needs a lot of oxygen to keep functioning. Behind it, we've got a round hole called rotundum. So it goes O-O-R-O. Optic canal, orbital fissure, foramen rotundum. Rotund means round. The round part of the Capitol building is called the rotundum. And then this oval foramen is called foramen ovale, which is almost easy. All right, continuing on, because we have so much left to look at here. Orbitosphenoid, allosphenoid, presphenoid, and behind here, attaching to the basioccipital, basisphenoid. And that's why in human anatomy, the sphenoid bone looks like a mess, because it's actually all of these bones fused together. All right, looking on the inside here, staying at the back, 3D. So there's our exoccipital, supraoccipital. We have a plate here that divides where the cerebellum is from the cerebrum inside the skull. That's called a tentorium. We don't see that in human skulls. You'll also notice this hole here. This hole corresponds with the hole on the outside here. Tympanic bulla, external auditory meatus, or canal. Sound waves come in here to be processed by the middle and inner ear. Now that information has to leave here and go to the brain, so it exits by way of the internal auditory meatus. To house the middle and inner ear, we've got to have the bone very, very dense. And so you'll notice that this bone in a lot of the specimens is lighter than the bones around it. It's because it's the densest bone of the skull. It's called the petros. Petros from the Latin word peter, which means rock. A petrified piece of wood is a wood that the wood tissue has been replaced by minerals, right? It's a wood that's become rock. So peter refers to the rock. All right, now this one shows it nicely. This is basisphenoid. Here we have what looks like a saddle shape. There's the back of the saddle. There's where the little guy's butt would go. There's the pommel. This part of the basisphenoid is called sella turcica, which means the Turkish saddle. The ancient Turks used to fight from horseback with lances and spears. You don't have a tall back to your saddle. When you spear someone, you're going to get pushed off the horse yourself, and then you get killed by the infantry. So this has a nice high back to it. Pommel here. Where the little Turkish warrior's butt goes is actually for an endocrine organ called the pituitary. Another name for pituitary is hypophysis. So this depression for the pituitary is called hypophyseal fossa. All right? Now we get to the part that's caused some of you trouble. We're in the nasal cavity, and we have our vomer here and our ethmoid here. It depends on how the skull was cut, how much you can see of that. This one shows it really nice. There's the perpendicular plate of the ethmoid. Here's the vomer. You notice the vomer rises up to meet the ethmoid. The ethmoid comes down to meet the vomer. Together, plus with cartilage, they form a wall that divides your nose into a right and left-hand portion. This is called the nasal septum. All right? But if the skull's not cut just right, since it's so thin, you're not going to see that at all. So the vomer is easier to see on the inside. And even though I've tried showing you pictures from different places, seeing it under here is really hard. The best place to see it from the front is looking into the nose, right there on the floor, right there coming up. All right? The best place really is to see it from a side view like this, a sagittal view. All right, so our ethmoid has a lot to do with the nose. Okay, this is the perpendicular plate of the ethmoid. If you take it off, we see all these scroll-like structures here and here. These are the ethmoid turbinates. They help to break the airflow into smaller passageways so you can more efficiently warm, filter, and moisten it. And in the case of many mammals, all of this is covered by olfactory receptor cells. So that's why they have a much better sense of smell than you do. The axons from those olfactory receptor cells enter into the cranium here. All these little holes are called olfactory foramina, foramina being the plural of foramen. Sitting right on here is the olfactory bulb. In fact, this is the impression of the olfactory nerve going into the cerebrum. All right? All of these holes are on a piece of the ethmoid called the cribriform plate of the ethmoid. You would have to be old people to be able to remember that easily, because there was a game older people used to play called cribbage, where you've got a board with holes in it, and then you roll dice, and that dice tell you how many holes your peg can move down. Whoever gets to the end first wins. So all these holes, cribriform. Okay, so whenever we see cribriform or cribba, that refers to a lot of holes. Okay, we'll see that with the kidneys also. All right, so one of the neat things is when you look inside the skull, you can see the impression the brain made on the inner aspect of the cranium, because the brain resides within the skull. But everything's 3D, right? So squamous bone, parietal bone, frontal bone, and in some cases, we can see an opening here in the frontal bone. That's the frontal sinus. Sinuses are found only in mammals. But when you drink a milkshake really fast and you get a brain freeze, it's happening right here. It's due to changes in air pressure within your frontal sinus. One last thing I didn't mention about the celoturcica, I'm sorry, but the celoturcica is part of the basal sphenoid. So if I put a tag here, I could ask 3 questions. Name this saddle-shaped structure. Name this depression. Name this bone. I told the first lab this. On the day of a practical, read the questions. It's easier for us to tag a structure and write up 2 sets of questions. So we just change the question sets between practicals. We can change the exam without having to spend hours retagging the specimens. If I write the 2 different sets of questions up ahead of time, quick, 2 minutes, boom, boom, boom. We got a brand new exam for you. All right? So reading makes all the difference on these exams. All right, and of course this one you can actually see a sinus within the sphenoid. That's the sphenoidal sinus. One of the ones that often has imperfections. Those of you who have upper respiratory tract infections that you can never shake, sometimes it's due to imperfections in here where the bacteria hide out when you're on an antibiotic regimen, and then they come out afterwards. And that's why you get an infection again and again and again. Okay, I think I covered all of that. Anyone have any questions?